Hello everyone. Welcome to the next lecture. Today we are going to talk about the lysosomes. So, as the name indicates, lyso stands for lysis. This comes from two words. This is a combination of two words. Lyso means lysis, breaking down something, to loosen something, and some comes from soma, that means body. So basically, lysosomes are the vesicles. These are the cellular organelles which are involved in breakdown of different products. As we have previously discussed, that most of the cellular organelles, they're composed of a lipid bilayer. So the same is true for the lysosomes as well. They're generally of different sizes and different shapes. They can be pretty small and they can be pretty large. I mean, the difference between the smaller ones and the larger ones could be like 10 times. So it appears they're of different shapes and different sizes. So it appears that they also might have different types of functions. Like some of them could be involved in extraction of nutrients from the debris or material that the cell has engulfed or absorbed. Some of them could be involved in killing microbes, while other could be involved in recycling or degradation of the older cellular components, while some of them could be involved in breaking down the debris. So they have different sizes and they're of different shapes, and therefore they might also have different functions. As far as their size is concerned, so they generally range from 0.1 to 1.2 micrometer in size. They're also known as suicide sacs or suicide bags because they're known to kill the cells themselves as well. So this is the reason we call them suicide bags. So generally these the lysosomes, they're spherical in shape and they contain specific enzymes, what we call acid hydrolases. Why do we call them acid hydrolases? Hydrolases means that these are the enzymes which are involved in hydrolysis or breakdown of the products. And acid comes from the acidic environment that these enzymes work at. So these enzymes work at a lower pH comparatively. So lower pH means this is lower as compared to the cytoplasm. So generally, here inside the cytoplasm, the pH is around 7, while inside the lysosomes, this varies between 4.8 to 5, and outside it's 7 to 7.2. So the outside of the lysosomes, in the cytoplasm, the pH is relatively alkaline or neutral, rather neutral, and inside of the lysosomes this is acidic. So these enzymes which are found inside the lysosomes, which are involved in the breakdown of the products, we will see later how do they break down the products, but these enzymes which are living inside the lysosomes, they work at an acidic pH, generally lower than 5. And this lower pH is maintained by specialized proton pumps. These are transmembrane proteins which keep pushing protons into the lumen of this, or into the inside of this vesicle, so that it keeps the pH lower. And generally there are two different types of transmembrane channel proteins which are involved in this. Transport proteins which are involved in this process in maintaining pH. There are these proton pumps which keep pushing proton into the inside of the lysosomes, and there are chloride ions which work as antiporters. So that maintains the lower pH inside the lysosomes, and these enzymes work inside the lysosomes. So this is also to the advantage of the lysosomes. Or to the cell, because if this lysosome ruptures and if these enzymes are released into the surrounding cytoplasm, then the cytoplasm or the cell remains protected from the release of these enzymes, because these enzymes work at a pretty low range of pH. So they work in an acidic environment, while the environment outside the lysosomes, inside the cytoplasm, is more neutral. So these acidic enzymes cannot work at neutral pH. So the cell remains protected from the effect of these enzymes, which would otherwise destroy everything. They're found in all animal cells, while in fungi, algae, and plants, the same role is performed by the lytic vacuoles. But in case of animal cells, they're not found in the red blood cells. And the reason why they are not found in the red blood cells, we know that the red blood cells, they do not contain any membrane-bound organelles. They do not have any Golgi apparatus, any endoplasmic reticulum, any lysosomes, any nucleus, etc. So I'm talking about the mature red blood cells. They do contain these organelles when they're during their in their premature stages, when they're being formed yet. But once they are mature, the internal content of the RBCs is generally replaced by hemoglobin. So they're like hemoglobin-filled sac-like structures which do not have any internal organelles. So for the same reason, they do not contain the lysosomes as well. The functions of the lysosomes are to ingest the materials and, as we have discussed, to destroy the older, obsolete cellular components, to engulf microbes and kill them and extract nutrients, etc. And they do so with the help of 50 different enzymes, which work at, as we have discussed, at a pH which is lower than 5. And they contain all the different types of enzymes. They contain proteases, which are involved in the, they contain lipases, which are involved in the degradation of lipids. They contain amylases, which digest carbohydrates or sugars. They contain proteases, which digest proteins, and they contain nucleases, which digest nucleic acids. So anything that goes inside the lysosomes is destroyed because of the presence of these enzymes. But one might think that how do they keep themselves protected from the effect of these enzymes? We know that they themselves are composed of the same lipid bilayer. They also tend to contain the proteins in their membrane. But if they contain if they have such an aggressive environment on the inside, how do they protect themselves? So they protect themselves with the help of an internal thick layer which is known as glycocalyx. And this glycocalyx is composed of special proteins, what we call LAMPs, LAMPs. LAMPs stands for lysosome-associated membrane proteins. And these proteins are heavily N-glycosylated with special sugars, what we call polylactosamines. So it means they are glycosylated proteins. And because of this heavy glycosylation, the enzymes inside the lysosomes cannot destroy these proteins, and they keep it protected. But we know that we have learned that lysosomes also contain glycosidases. So glycosidases would, one might think that they would try to destroy the glycocalyx. They definitely do so, and this is the reason that lysosomes do not live inside the cells forever. So they live, they have got like the enough number of safeguard mechanisms to protect themselves for a specific period of time. And after some time, the lysosomes themselves also become old and they also get destroyed. The important thing is here is the number of lysosomes destroying at the same time inside the cell, and how fast do they do so. So if they do not destroy too fast, then it's like there is one lysosome breaking down, and after some time another lysosome is breaking down. Then the cell has got enough time to repair itself if any of the damage happens to the cell because of these enzymes. Although these enzymes would not be very efficient outside in performing their functions outside these lysosomes because of the neutral pH of the cytoplasm. But these lysosomes are not designed to live forever, because if they do so, we know that they also kill the microorganisms. If they do so, the lysosomes, the way lysosomes have evolved to protect themselves, the microorganisms could also hijack their protection mechanisms or safeguard mechanisms to kill the cells. I mean, like, they have got these enzymes which can kill the cells, and they have got this glycocalyx to protect themselves. So microbes also tend to evolve. So this is to the advantage of the cell itself, that they live for some time and they get destroyed. They do not have the strategies to protect themselves forever. Another important thing is that they get destroyed after some time, because the cells also produce some some products which are meant to be protected from the lysosomes. Means they cannot get destroyed by the lysosomes. If these products are engulfed by the lysosomes, and if they are not destroyed by the lysosomes, and lysosomes themselves do not get destroyed, then these products, which cannot be destroyed by the lysosomes, they would keep accumulating inside the lysosomes, and they would take up a precious space inside the lysosomes. So this is necessary that lysosomes themselves also get destroyed after some time. But if they do it one by one, not like all the lysosomes getting destroyed at the same time inside the cell, the environment of the cell, that is the cytoplasm, it can neutralize these acid hydrolases and protect its other organelles. So the number of the lysosomes getting destroyed at the same time, and the rate at which they die out, that matters. So there's some sort of balance in this case, that they do get destroyed, but not at such a high pace, or not at such high numbers that they destroy the cells. So this is how they protect themselves, and they get destroyed, and the cell is also protected. The enzymes that they contain, they're specialized enzymes, and these enzymes, they're synthesized, they have got their genes inside the nucleus, so transcription takes place, and their mRNA. The mRNA for these enzymes is transported to the nucleus, and from the nucleus it goes to the ribosomes. Ribosomes translate it, and then these enzymes go to the as we have previously discussed, these enzymes go towards the, let me go to that picture, please. Yeah. So the mRNA is produced, and then these proteins are translocated into the I think it should be a post-translation mechanism, this translocation. So the mRNA is the mRNA. So this is the mRNA for the different genes, or for the different enzymes which are found inside the lysosomes. So the genes transcribe the mRNA is translated into the protein is translocated into the endoplasmic reticulum, and from the endoplasmic reticulum this is sent to the Golgi apparatus here. So they're produced in the ER, and from the ER they go into the Golgi apparatus. But before they enter into the Golgi apparatus, these enzymes are tagged with some sort of tags, what we call mannose 6-phosphates. So these mannose 6-phosphate molecules on these specific enzymes, they indicate that these enzymes need to go to the lysosomes. So what happens that when these enzymes, they get tagged with mannose 6-phosphate, the Golgi apparatus in its membrane has got receptors for mannose 6-phosphate. So with the help of mannose 6-phosphate, these enzymes, they get embedded into the vesicles with the help of receptors. So they remain bound with the receptors. And then these vesicles are transported from the Golgi apparatus towards the endosomes. We will study this later. Let's go back to the lysosomes. Yeah, so let's talk about them here. But one thing I would like to mention here is that sometimes there's some damage to to these genes which are involved in the production of these acid hydrolases. So, and they can lead to roughly 30 genetic disorders which are collectively known as lysosomal storage diseases. And what happens that inside the Golgi, so the enzymes have been produced inside the nucleus, the mRNA goes into the cytoplasm, it gets translated um, into the cytoplasm, and the protein is translocated to the endoplasmic reticulum. From the endoplasmic reticulum, the protein is translocated to the Golgi apparatus, and from the inside the Golgi apparatus, they're associated with their receptors, and they are transported. They're transported in certain specific vesicles, and they fuse with the endosomes. We will discuss this later, what happens afterwards. So here are the three different pathways which deliver materials for lysosomal degradation. So one is phagocytosis. I think you're pretty familiar with it. Bacteria or solid particles are engulfed by the cell, and then we have got pinocytosis, which helps the cell engulf suspended particles. And then we have receptor-mediated endocytosis, where particular specific products are internalized when they bind to specific receptors on the cell membrane. So all the three processes basically lead to the formation of vesicles. And these vesicles are known as endosomes or phagosomes. So in case of endocytosis or pinocytosis, we call them endosomes, and in case of phagocytosis, we call them phagosomes. And ultimately, they fuse together to constitute what we call late endosomes. And what happens, how do these endosomes develop into lysosomes? We have previously discussed that the enzymes are contained inside the vesicles which bud off from the Golgi apparatus. So Golgi apparatus sends these vesicles, and these which contain the enzymes. So here's the vesicle, for example, and this vesicle contains enzymes which are responsible for degradation of the products, and these are mannose 6-phosphate associated enzymes. So this vesicle basically fuses with this endosome, and when it fuses with the endosome, the inside of the endosome is slightly acidic, and because of this acidity, what happens that the mannose 6-phosphate tag is removed from these enzymes, and these enzymes become functional. And fusion of these vesicles with this late endosome, so vesicles keep fusing with this endosome, and they keep pouring their enzymes into this endosome. This endosome later develops into a lysosome, and the enzymes which were poured into this endosome by the vesicles which are coming from the Golgi apparatus, these enzymes start destroying the content which is inside the lysosome, and this is how a mature lysosome is formed. So these are two extracellular mechanisms, or rather three mechanisms: phagocytosis, pinocytosis, and receptor-mediated endocytosis, which help the cell engulf particles which come from the outside. So this is the reason the lysosomes are also known as the stomach of the cell, because they help in the digestion process of different products. We have also discussed that lysosomes are also known as suicide bags. So because if they all of them burst at the same time, they can kill the cell. But at the same time, they're also involved in killing or degrading or recycling the old components of the cell itself. For example, here we have got a mitochondrion. It is old now, it's obsolete, and it cannot perform its function normally. So the cell cannot keep up accumulating different organelles here. So time to time, the cell needs to recycle them. Means the older ones are destroyed and the newer ones are synthesized or formed inside the cell. So how do they get destroyed? This is done by the lysosomes. So in autophagy, this is what we call autophagy, means eating itself, or self-digestion. Autophagy means the cell eats up its own components. So what happens, for example, this is a mitochondrion which needs to be destroyed. Some smooth endoplasmic reticulum wraps around this mitochondrion and makes up an isolation membrane which isolates this mitochondrion from the rest of the organelles. So this mitochondrion is wrapped around by a membrane, and we know that the endoplasmic reticulum is also composed of a phospholipid bilayer membrane. It means that the composition of this isolation membrane and the membrane of the lysosome, this is identical. And if the two membranes are identical, this is known as autophagosome because it has solid content itself inside it. We call it a phagosome, and autophagosome because this is the cell's own component. So when this is engulfed, this membrane wraps around the mitochondrion. This is more like a phagosome, which has got a solid particle inside and a membrane outside. And the structure of this membrane is identical to the structure of the lysosome. Means these two structures can fuse with each other. And this mitochondrion is poured into this lysosome. And then it gets destroyed by the hydrolases that this lysosome contains. Another thing that can happen is that sometimes these endosomes, multiple endosomes, can fuse together with each other, and they constitute what we call multivascular bodies. And when these these are multivascular bodies which can which get originated because of the fusion of all the different vesicles, whether these were like phagosomes, these were endosomes being formed by the cell through the process of pinocytosis, receptor-mediated endocytosis, or phagocytosis. So multiple smaller endosomes can fuse together to form giant multivascular bodies. We call them multivascular bodies because even inside them they can contain some vesicles once again. So this is a vesicle itself, and it contains certain other smaller vesicles inside it. And here we have got a lysosome. So these multivascular bodies can also fuse with the lysosomes, and now lysosome has got all the different things inside it. It has got mitochondrion, which came from the autophagy. It has got some suspended particles which came from the from the pinocytosis. It has got some receptor associated or receptor bound products which ended up here because of the receptor mediated endocytosis. And even this lysosome can pick up other products, so and from within the cell, and which were the products of the cell itself or components of the cell itself. Now here we have got autophagy, and this is again of two types. One is macroautophagy and the second one is microautophagy. So macroautophagy means these are the bigger cellular components which are eaten up or internalized like phagocytosis into the lysosome. And lysosome can also engulf endogenous proteins or suspended particles in a fashion identical to pinocytosis. So these suspended particles, smaller particles, they can be engulfed by the lysosome, and lysosome can digest all of them with the help of its enzymes. Okay, I think this is sufficient for today. Thank you very much for your attention. If you have any questions, you can post your questions, and I would be glad to answer them. Thank you very much.